Hello, I'm Harry Glorikian, and this is The Harry Glorikian Show, where we explore how technology is changing everything we know about healthcare. It may feel like generative AI technology suddenly burst onto the scene over the last year or two, with the appearance of the text-to-image models like Dall-E and Stable Diffusion, or chatbots like ChatGPT that can churn out astonishingly convincing text thanks to the power of large language models. But that's not really true. As with most every technology revolution, the real work has been happening in the background in small increments for many years. One demonstration of that comes from Insilico Medicine, where my guest this week, Alex Zhavoronkov, is the co-CEO. Since at least 2016, Alex has been publishing papers about the power of a class of AI algorithms called Generative Adversarial Networks, or GANs, to help with drug discovery. One of the main selling points for GANs in pharma research is that they can generate lots of possible designs for molecules that could carry out specified functions in the body, such as binding to a defective protein to stop it from working. Drug hunters still have to sort through all the possible molecules identified by GANs to see which ones will actually work in vivo or in vitro. But at least their pool of starting points can be bigger and possibly more specific. Alex comes from a background in computing rather than biology, and he says that when Insilico first started touting this approach back in the mid-2010s, few people in the drug business believed it would work. So to persuade investors and partners that the technology works, the company decided to eat its own dog food, so to speak, and take a drug designed by its own algorithms all the way to clinical trials. It's now done that. This February, the FDA granted orphan drug designation to a small molecule drug Insilico is testing as a treatment for a form of lung scarring called idiopathic pulmonary fibrosis. Both the target for the compound and the design of the molecule itself were generated by Insilico's AI. And with the orphan drug designation, which supports the development of drugs for rare diseases, Insilico is now in line for potential federal grants, tax credits, and extended market exclusivity, assuming it can get all the way to FDA approval. The designation was a big milestone for the company and for the overall idea of using generative models in drug discovery. I asked Alex to come in and talk with me about how Insilico got to this point, why he thinks the company will survive the shakeout happening in the biotech industry right now, and how its suite of generative algorithms and other technologies, such as robotic wet labs, could change the way the pharmaceutical industry operates. Here's our full conversation. Alex, welcome to the show.
Very happy to be with you, Harry.
So, as I was just saying, it's been a while since I've seen you. I remember when you were telling me about Insilico, I feel like it was 2015, 2016, definitely way pre COVID, but Insilico has so much going on in so many areas. I mean, I'm trying to frame this for the people that are listening, but for example, I know you provide AI drug discovery tools for other companies, but you also have a pipeline of your own drugs that you're developing. So I was hoping maybe that you could start by telling us, how do you boil down a description of the company for people who are not, say, familiar with it, right? Is there a single technology or theme that unites everything that you do?
Sure. Well, again, it's a great pleasure to be speaking with you again on record today after so many years, because 2015, 2016, it was even difficult to explain what generative AI was. And right now, the consumerization of that technology made it very easy to explain, right? Because you remember, I was probably showing those flowers where you say, okay, add one more petal, here is a petal. And we can do the same with molecules or synthetic biological data. And people did not get it. And many of them criticized this technology and this approach. I know some of them still do. what unites Insilico currently, both the services and the internal pipeline, is patient first. So we are very interested in delivering drugs to the patients as quickly as possible, either through software or collaborations or through our own internal pipeline. So about 2 years ago, when I first met my co-CEO, well, 3 years ago now, when I first met my co-CEO, Dr. Ren Fang, or Feng Ren, depending on where you are. PhD from Harvard, 11 years at GSK, and then he listed a company called Medicilin. It's a big contract research organization which competes with WuXi AppTec. And at that time, it was $8 billion. Once he saw what Insilico could do as a provider, right? So we started synthesizing our chemistry at his company, and he saw that, and it was pretty magical at that time. And he decided to join the company, right? And said that, look, I don't want to provide services. I want to discover and take advantage of this technology, which is pretty much like, as a consumer today, if you see how ChatGPT performs, right, some certain tasks, you also want to get into the field. And back then we thought about, okay, what is a central theme of Insilico, just like what you've just asked, in the context of very rapidly changing technology, because technology is changing much faster than we think, than we see. So by the time I finish talking, somebody is going to publish a generative AI paper somewhere that will have some impact. And at the same time, drug discovery is very slow, right? So once you start the experiment, very often you need to wait for a very long time until it completes. So the pace of innovation in AI is, of course, much faster than the pace of innovation in drug discovery. So how do we cope with that? And we thought, well, let's forget about technology and drug discovery, and let's think about who we are and why are we here for, and let's come up with a set of values that will be with the company for as long as it exists. And we want to be in this game for a very long time. As you know, we're also focusing on longevity. And those values we actually brought a team together. So we had an offsite event with all employees, and we decided that we exist for 3 purposes, and we have 3 fundamental values. One is patient first. So whatever technology is out there, whatever the investor interests are out there, whatever stakeholders are out there, the most important stakeholder is the patient. And we need to figure out a way to deliver the drug to the patient as quickly as possible, regardless of the means. So second is relentless innovation. So we want to ensure that we do not stop innovating, regardless of the pace of change in technology. And right now, we saw several cases where our software started performing so consistently well that people get addicted to it, and we don't want to change the engines, right? And it's kind of what happened to Google recently, right? You get addicted to Google, you still will search Google, but with the advent of generative tools, you now want to change, right? And in our case, we really want to see those generative tools and many other tools integrated into the platform, so you cannot stop. So what our fundamental value is relentless innovation. We will not stop. That's why we have 2 CEOs in the company, right? Just to facilitate for this value. And the third value is transparency and integrity. So we want to ensure that everything that we claim is backed up by facts. And we try to have a few research papers usually, so academic papers supporting the claim. So 2 or 3 peer reviewers from academia or industry would be able to back this claim because it's peer reviewed. And those are the 3 fundamental values, and they basically define what the company is and what we do. And that's why we have 2 CEOs. So Dr. Ren, after he joined as a chief science officer, but then very rapidly demonstrated that he can outpace pretty much any other biotech company, right? So we nominated 2 preclinical candidates within 8 months after he joined the company. So that's pretty cool. And last year we nominated 9 preclinical candidates, so 2022. Just for reference, big pharmaceutical companies, if you look at just small molecule drug discovery and just fully internal R&D, they usually nominate like 4 to 5 to maybe sometimes 10 if if it's the best year in the world in history, but usually it's less. And it basically means that on a very tiny fraction of the budget, we managed to perform at the level of a big pharma company. And we tried to go after more novel targets. And Dr. Ren became a co-CEO. So we decided that, okay, we're going to have 2 heads. One is going to be AI to facilitate for relentless innovation in AI and also robotics. And the second head is discovery and also contribution to AI from the drug discovery perspective. So that is to answer your question. So how did we develop in software and development and discovery at the same time? So we have 2 CEOs. I'm in charge of tech, and Dr. Ren is in charge of the classical kind of backbone of Insilico, where the value is created by going after new targets or commercially tractable targets and doing your own drug discovery and development.
So to jump on that, or to build on that, right? You guys got some exciting news in February when the FDA granted Insilico orphan drug designation for a drug you're testing as a treatment for idiopathic pulmonary fibrosis. And I believe it's called INF018_055. I love all the code names we give our drugs, right? And my understanding is that you discovered it using your own AI for finding small molecule drugs. And it's one of the ones that's farthest along in your pipeline. So could you tell me the short story behind that drug as a way of illustrating the company's work overall?
So sure, will do. It's kind of sad that I cannot use the visual aid to share a screen. But basically, in around 2016, we published our first research paper where we demonstrate the use of generative adversarial networks for drug discovery. We then, for several years, perfected that system, and in parallel, we also worked on generative biology and all kinds of deep neural network approaches to identification of novel targets. And in 2019, we started releasing the software to the market. And then we figured out that, look, now if we want to go after something big and also to promote our software, we need to make a big demo of how the software would perform in real world context. And we also noticed that many users of the software were very skeptical. They usually do not want to bet on newer targets or synthesize more interesting molecules. And they needed to have a real life demo. So we raised some significant amount of funding. Originally, it was 37 million, and decided to go after our own project and our own drug discovery. So we decided to go after, my passion is aging. So I wanted to find something that has dual purpose, so age related, but at the same time commercially tractable and purposable in a specific disease. So I decided to go after fibrosis, specifically as a biological process. It's not a disease by itself, right? You need fibrosis when you are younger, or when you need to heal some wounds, or just have a patch in a tissue. But when there's too much of it, when there are too many patches, systems break, right? And you want to ensure that there is no excessive fibrosis. So we decided to go we linked it to aging. So we built massive models of aging. back in 2016, 17, using gene expression data and other data types, found a few targets that are implicated in aging and fibrosis. And then we create quite a bit of context, so biological context behind those targets. We prioritized 20, we could test only 5. All 5 worked, and we prioritized 1. And then we decided, okay, well, we now have a demo to show, well, do we want to release it right away? And at that time, we already started publishing a bunch of other things for target discovery, many proof of concept. And we noticed that people are not necessarily, buying into this early stage research and using the software more or trusting the software more. We needed to go further. So we thought, well, why don't we also use our generative chemistry AI, which can generate small molecules with the desired properties. So for that specific target. And why don't we continue this into the real therapeutic program? So we synthesized a few molecules, tested, got really nice nanomolar inhibitors, and then we optimized several times, got those molecules to be very advanced, so basically to preclinical candidate. And we did it on only a few rounds of synthesis. We synthesized just 79 molecules for that program. And most of the molecules were hits, and many were amazing hits, right? So single-digit nanomolar and selective, and with many other properties, orally accessible, metabolically stable, basically up to 32 properties were met. And we did a few efficacy experiments. So in liver fibrosis, in kidney fibrosis, in lung fibrosis, and skin fibrosis. And in lung fibrosis, we've managed to achieve a preclinical candidate for idiopathic pulmonary fibrosis. So we did multiple experiments in human cells, patient-derived, cell culture, then several mouse models. Usually those were bleomycin-induced mouse models. So you induce fibrosis and then show how you rescue it with your own drug. And we did this very, very quickly. In parallel, we have achieved a preclinical candidate for kidney fibrosis and also almost achieve a preclinical candidate for skin fibrosis. And when we thought, okay, well, will this be enough for people to believe that AI is outperforming humans? Not outperforming humans, but outperforming the traditional methods. And we did a quick survey and realized that, okay, well, most of the companies would like to see some clinical data. So we decided to go all the way and did IND enabling studies. So GLP talks and the entire IND package. We did it reasonably quickly, but there you cannot move quicker than the contract research organization can provide you with the data. You have to follow all the protocols, right? Because the FDA will require it. And we actually did a little bit more because we wanted to have several redundant data packages. And since the target was new, we decided to also do a small pilot phase 0 study in humans. That was November 2021. And we completed a very small 8 healthy volunteer study in Australia, looked at how the compound is distributed, did very, very basic safety. And then we decided to go phase 1, full phase 1, so 80 healthy volunteers, and started it and announced it in February 2022. And just recently, we've announced the completion and that we received the top-line data from this phase 1. So we are hopefully going to start phase 2 soon in the US and in China. And then we applied for the orphan drug designation and got that from the FDA. So we're very, very happy about that because it provides a lot of benefits for the program, including it becomes more investable, right? Because this Inflation Reduction Act has many negative effects on small molecule drugs, right? So companies do not really have a lot of luxury to invest in innovation. And we are probably the most innovative program, in my opinion, out there, period, right? And the fact that we got ODD is great, right? Because now we can say that there are additional benefits associated with this program, and maybe we can have longer longer market exclusivity after approval. So that's the major benefit from the ODD.
So, you almost have to thank those guys that didn't adopt the technology earlier to force you sort of to go down this path, right? It's the Clay Christensen innovator's dilemma, which is they're risk averse and they say we've got enough money that we're willing to wait, but that pushes a few companies along to really, and at some point you may not even need them, right? Because of the engine that you've created. But that sort of brings me to.
We're not going to talk about that. We need them. We need big pharma very, very much. Somebody needs to sell the drugs, and somebody needs to do the review of the data. So yeah.
So I want to go back to 2014, right? The company got started. What did the market need? What opening did you see? I mean, what were the unique technologies that you had developed that you thought could address those needs? And I mean, those technologies have advanced, orders of magnitude since 2014. I remember looking at it back when we were first talking about it compared to where we are now, and they're not even. I don't even know how you draw the curve for how much it's changed, but walk me through the beginning days of like, what did you see? What did you start with? And how are you constantly innovating to keep up with this? Because this is the big question for every company.
So sure. My background is in GPU computing, right? So computer science, and then I worked for a GPU company for a number of years. And that gave me a little bit of a competitive edge when the deep learning revolution started in 2014. So back then, I was already in biotech. So I switched into biotech from GPU computing around 2004. So 18 years ago, well, 19 years ago. In GPU computing is necessary for deep neural networks, right? You have GPU, general purpose graphics processing units, that allow you to perform many highly parallel computational intensive tasks much better than those CPUs. So most of the deep neural networks in existence today, they are trained either on GPUs or on specific semiconductors that are made for deep neural networks. Now you have those as well. But previously it was GPUs. And after I quit the semiconductor business and decided to focus on biotech and did my grad work at MSU, did my grad work at Johns Hopkins, worked in the industry for a while. A lot of my friends from ATI Technologies migrated into NVIDIA. And NVIDIA, as you know, is the technology giant, which is the leader in anything GPU, anything AI. So around 2013, one of my friends called me up and said, well, you know what, Alex, you should come in and give a talk at NVIDIA because, you are in deep learning, right? And you are in GPU. We're a GPU company. We want to expand into life sciences. So I think that at that time, so Jensen Huang, the CEO of NVIDIA, is probably the most visionary technologist, entrepreneur, and just beautiful human being who built this industry. He already back then thought about the applications of deep learning to healthcare. And they wanted to learn from the experts. So I came in, I gave a talk 2014, and 2015, we started participating in those NVIDIA GTC, ETC competitions, became a finalist. At that time, most people didn't get what we were doing, right? Because we were all also trying to, whatever I do is focused on aging, because I think that's the ultimate killer and ultimate biological process that leads to many diseases. And if you don't understand aging, you probably wouldn't be able to understand many diseases as well, right? So you're just going to be looking at individual targets and some very primitive mechanisms without, understanding the global picture. And our idea back then was to train deep neural networks, massive deep neural networks, to predict human biological age using any data type, and using age as the feature that we predict, right? Because we change in time. So the idea was to look at biology in time. So it's actually pretty credible. But at that time, I didn't know how to pitch it properly. So we struggled for a couple of years to fundraise, because back then, nobody would want to invest in an idea like that. Now the industry actually kind of emerged, that is longevity industry. But back then, you would not be able to fundraise for something like that. So I had to put everything I had myself. So I even sold my apartment and put the money into Insilico to demonstrate that deep neural networks can not only outperform traditional approaches, but it's a completely different way to do things, period. Right? So we actually, back then, I started hiring people, and many of them demonstrated that, gradient boosting learning is outperforming deep neural networks in certain tasks, or support vector machines are great for certain tasks. But I decided back then that, look, we're actually going to use deep neural networks, period, right? And just bench when we use something internally has to be deep neural net based. It's not, doesn't make sense, but trust me, in the future it will, right? And turns out it was a very good bet, because once generative adversarial networks were published by Ian Goodfellow, and that was 2014, together with Yoshua Bengio. So that was the cusp of the deep learning revolution, right? So first, basic deep neural networks, convolutional deep neural networks, and feed-forward deep neural networks started outperforming humans in image recognition, voice recognition, text recognition, many other tasks. And generative models got published. Once they got published, I decided to immediately jump onto the new technology, and we wanted to use GANs everywhere, right? So if you look at the literature, media, whatever, we met back then, I was already preaching GANs, right? And we thought that, look, we are going to use GANs and to try to generate synthetic biological data, small molecules with the desired properties, try to forecast into the future some clinical trial designs and clinical trials. Also generate entire human populations using age as a generation condition. And back then, a lot of people just didn't get it. So we published some really cool papers, filed some really cool patents. So that's actually another story I can talk about. But investors, serious investors, didn't get it, right? And only in 2017, when I first synthesized the molecules and tested them at Wuxi Aptech saw the results. So they saw real chemistry. And they said that, look, it's actually pretty primitive now, and some of the molecules are not synthesizable or good. But after testing, they got convinced. They invested in Leather Series A in 2018. So that's when we actually started getting capital attention. And with Wuxi's support, once they joined the board, they basically introduced a lot of quote unquote big boy features into the company, right? So we became a much more credible player in drug discovery, right? Because Wuxi also carries a lot of weight, and they have experienced drug hunters, medicinal chemists, huge number of synthetic chemists, biologists. So we piggybacked on the company quite a bit. And later, once we decided to introduce the software, we of course got a bunch of other investors who wanted to make it happen and also deliver this software to the community, so it's good for everybody. And coming back to 2014, we never imagined where we're going to be today. But the bet on relentless innovation and constant change was a very good bet. That's why, again, we have two CEOs today, so I can continue doing what I was doing and being, quote unquote, the AI technology cowboy. This attitude is very often, people don't like this kind of attitude in
Yeah, I
pharma.
know.
Yeah, especially
I
when
know.
you're
I have
making
people around me.
statements. Yeah,
I know I have people around me like this all the time. And I don't, because the turns of technology are becoming so compressed that if you're not looking and not moving, I don't think people fully appreciate how quickly things are changing. And sometimes I can't read enough or fast enough to keep up with what's happening to sort of be able to draw a line of where the puck is actually going.
exactly. And at that time, we also faced a lot of resistance. So, some of the people who I deeply admired, I still respect them a lot. But back then, I admired these people. They were like heroes of mine, right? They built drug discovery companies based on computational approaches, right? Of the of the past. We actually still use those approaches even today, and they work. They're just slower, and you can combine them as in your big Lego constructor. You can still fit them in, but they are not the main driver of our engine. So many of those former heroes of mine, they actually started criticizing what we do, right? So because in drug discovery, very rarely you get the drug on the market, right? And very rarely you get the drug in the clinic from computational approaches, right? So that makes a lot of people very skeptical, and there are only a few key opinion leaders who actually have done that. So have taken a drug from the computational stage all the way into phase 2 clinical trials, uh, even for known targets. For something that is novel, there are I know only a couple, and back then many of them started criticizing generative approaches, right, and saying that, look, it's an incremental change, it's not going to be a paradigm shift anywhere. Show us the drugs in phase 2 or past phase 2. You're going after two easy targets. The molecules do not look novel. You cannot replicate that easily because the model is trained, right? And we don't know what's in the data set. And you need to dumb it down to like one primitive model instead of a huge Lego system to demonstrate, that small piece of Lego can actually do magic. No, it can't. And we struggled quite a bit. And once those people started openly criticizing and even writing papers on that, I got really frustrated, right? And now, of course, I still respect them, but they're not my heroes anymore. What I think is the best criticism is a demo of superior performance with your own model, right? So we try to compete on the outputs of technology and technology itself, and demonstrating that you can develop better tech, rather than trying to criticize somebody who came up with a better tech, even if the tech is still uncertain, right? And you see, so one of my role models is, for example, Elon Musk, right? So you cannot take it away from him. 5 companies, 2 of them are delivering at scale, right? So you are driving his cars, you are using his satellites for communication, right, and with payload delivery. So, but very many people still criticize, saying, it's a forest fraud, bluff, and that will continue, especially people who are in the industry for a very long time, and they might say that he piggybacked on the government to get where he is today. Innovation is incremental. You get these statements all the time, and you won't be able to get around it. Those people just need to. Well, that's where aging actually is beneficial for a society. So yeah, so they won't get our treatments if they live to see them. But yeah, I was very frustrated, and we had to demonstrate clinical stage assets and also go after very difficult targets to prove them wrong in the investor's eyes, right? Because your main source of existence in this industry are the investors, right? And if your investors are people who have been in this industry for a very long time, and they of course see this criticism from those people who are heroes of mine, right, and they are very well respected, very often they also have doubts and questions, and they have to come up with their own teams to evaluate the tech and the performance of your drugs and the performance of your technology and software. And you actually need to show how the system delivers. They need to look at the molecules. They need to look at the algorithms. But it's a little different than publishing a research paper, because they can use the software, right, that you provide.
Right.
And it made our lives more difficult, this criticism. But after we demonstrated the experimental validation on more difficult targets and significant speed ups in more common applications, and then our partners generated, synthesized, tested, and got into late stages, investors actually like to tap each other on the shoulder and say, yeah, well, we knew it was great. Right? So now generative AI has been consumerized, right? And of course, a lot of people are criticizing the systems. Even myself, I basically published an opinion paper recently saying that, look, just generative AI, will love it, but don't use it for self-medication, for example, right?
Right.
So people just need to, so physicians need to advise their patients that, look, some of those generative systems are outstanding, you are relying on it. However, don't use it for self-medication. Today, I actually talked to my head of communications, and she's saying, Alex, you know what? We went to ChatGPT and asked, which company was the first company to use generative AI for drug discovery, and it shows us to be the first one and the main one, and says all those positive things. And I'm like, do you realize what you are doing? You are now utilizing ChatGPT as your primary source of information, and you're very happy about that? I mean, I'm very happy about that, but if you are quoting that as the ultimate arbiter, I think that you should be probably, doing a little bit more research into why it got into this, gave you this argument. And I'm, of course, very happy that it does. But I think that if people rely on those generative systems at the consumer level too much, much of this output needs to be benchmarked, validated, and we need to ensure that at the patient level, we do not cause any harm.
So, Alex, I want to get into the two tools you guys use. Your pharma AI platform consists of two different parts. One is, I believe, called Pandaomics, and the other one is called Chemistry 42. I'm wondering if you can walk us through, what each of those are designed to do. How do they complement each other? I mean, maybe you can start with pandaomics, and then I want to dive deeper into the chemistry 42, right? Specifically because it uses a generative AI approach. And as you've mentioned, like lately, GANs have been, oh my God, the rage, right? In tools like Dall-E and Stable Diffusion in art and ChatGPT in natural languages, right? I want to understand the two systems, and then what are the similarities and differences are between chemistry 42 and those other forms of generative AI that we've been hearing about.
Sure, there are actually three systems that we have.
Okay.
So there is pandaomics, chemistry 42, and Insilico. So, and all of them are using generative AI in one form or another. Chemistry 42 is most generative, so to speak, right? So you can trace the papers back to 2016. So I'll walk you through those three systems. So pandaomics, it's a system which allows you to identify novel targets for a broad range of diseases. We perform best in tissue-specific diseases where large amounts of omics data is available. So what this system does, it utilizes different data types. So either biological data types like gene expression, whole genome sequencing, and methylation. So those are the three predominant data types that the system works with, to come up with novel target hypotheses based on more than 60 target discovery philosophies. So you can come up with a target if it's genomically driven, right? So if there is genetic evidence that the target, that the gene is implicated in disease and it's somehow causal, this system has a philosophy for that. If you rely more on expression, so relative levels of expression of a certain gene change between norm and disease, or between different types of disease, the system will be able to capture that and also use it as a way to rank the target and implicate it in a disease. And there are many, many others, right? So again, more than 60 philosophies. And you only acquire those philosophies over time by working with key opinion leaders that discover targets, or big pharmaceutical companies, or biotechnology companies, or venture capitalists actually as well. So many of them, they have different ways to explore the target space. And this system, in addition to omics, it also has massive amounts of text data. So we estimate that the system sits on around $1.5 to $2 trillion worth of data. Because the way we capture this data, we track biomedical grants. And you can trace my publications on that to 2011. So by the time the grant is published, or given to the scientist, you've got an abstract for the grant and some basic information about the idea, the concept that the scientist is working on. We usually link this grant to publications later on, to patents, and then to clinical trials, and then marketed products, and even some real-world evidence. So that's what we do in a very careful, curated way. And we try to trace the omics repositories that stem from this grant as well in publicly available databases. And this ability to look at the propagation of knowledge creation and data creation from the time you get the grant all the way to the market product is the core of what Insilico is in general. So we look at everything in time. So we acquire data in time as well. And we look at the biological data in the system as the primary source of novelty for targets, right? Because many targets are out there, they're very well known, but they're very risky, right? So the more novel the target, the higher the risk. And then we look in the literature, patents, and many, many other data sources for more pockets of evidence. So there are different AIs, including generative AIs, that start building knowledge graphs and knowledge target dependencies and different causal models on trying to implicate this target in a disease further. And of course, if the target has been clinically proven to work, that's very validated, but it also is demonetized, right? So you should not develop chemical matter for something that is extremely well understood, and there are lots of good molecules against it, right? Investors just won't give you the money for that. So Pandaomics allows you to make those decisions on which target is implicated in disease, what is driving the disease, and also even some commercial tractability, whether you should proceed with this target or not, if you want to build a drug discovery pipeline for a specific therapeutic area. So Pandaomics solves that, and it's so easy to use that the high school students can use it. And they do. So we have cases where high school students from Norway and the US used Pandaomics to identify targets in diseases like glioblastoma and age-associated conditions, and presented posters. And now I know that one of the groups submitted a paper for the first time with authors going, as early as 16. So imagine back then, what were you doing when you were 16, right? I remember I didn't know basic biology.
I wasn't doing this now.
And so it gives students superpowers. And then Chemistry 42 allows you to prosecute those targets that you identify using Pandaomics with novel chemistry. And that's a very cool generative system, which can take either a ligand, so already known molecule, and then redesign it with desired characteristics. So pretty much, if you were to think generative in imaging, imagine if you got a picture of Brad Pitt, but you wanted to see Brad Pitt when he is 80, female, and with glasses and with some Asian characteristics. And a generative system would be able to produce a wide range of Brad Pitts with those features, right? But still retaining some features from the original template. So we can do the same with chemistry. Then, but that's a very rare application of our tool, mostly used for lead optimization. So when you already have a starting point and you want to make it better, that tool is for you. That pipeline in the tool is for you. The most common application is when you start with a target. So you give it a crystal structure. So you crystallize the protein, you identify a pocket within this protein where you want the molecule to bind, or the system does it automatically for you. Then you annotate this pocket, or the system automatically does it for you in the autopilot mode. And then within this pocket, the system generates molecules with the desired properties. And those properties you select. So pretty much like you would say in natural language to the system that I want to see a drug-like molecule that inhibits this prostate targets, binds to this active site or some other site on the molecule or in the protein with this affinity. And at the same time, it must bind only to this target and not anything else, or bind something else very weakly. It needs to be penetrable to a specific tissue, for example, cross blood-brain barrier, and it needs to be metabolically stable. And we want to ensure that you can take it orally, right, not inject it. So it needs to be stable enough for your gut not to digest it too quickly, or your liver to digest it too quickly, and many other parameters. So there are many parameters, so huge number of parameters that you can select for your molecule. And then the system starts generating using many generative engines. There are more than 40 generators in there using different approaches. Either it is GANs, or it is transformer neural networks, or it is genetic algorithms. And they all start generating molecules with desired conditions. And then we have a massive predictive system. So where you have more than 500 different ways to evaluate the molecules that are being generated by the generative system and evaluate them whether they're good or not. So using different approaches. In 2D, in 3D, if you have a little bit more time and money, you can also evaluate them using other representations of molecular structure, even go down to the physics layer. This predictive system is either rewarding or punishing each individual model, generative model, and the ensemble. So you've got the reinforcement learning system going on, right? So if the molecules that are generated by the system with the desired properties are bad, so they might be toxic, they might be not synthesizable, they may have many other properties that you don't want to have, the model gets punished. And it learns over time to generate really good molecules for that specific target with the desired properties. And at the end, you also can use this predictive system to rank the molecules that at the end are generated by this ensemble and say, okay, I want to synthesize the top 10. Or no, I want more diversity. I want to synthesize some molecules with this and this and this scaffold. And you can filter out and then send them for synthesis. So in the first pass, you usually generate and synthesize about 20 molecules. And many of them would be hits, depending on the target type and depending on how lucky you are or what your generation conditions or filtering conditions are. So you can get way outside the known chemical space with Chemistry 42. You now don't need to screen the libraries to identify the molecules that you want. You can generate a lot of those great molecules from scratch. So instead of searching for a needle in a haystack, you can create a haystack of great needles and then prioritize them for your tasks. And then we have a system called Insilico. This system actually incorporates some engines from Pandaomics, and it's predominantly used by, and we are piloting currently with many hedge funds and banks and capitalists who actually bet on small and medium type biotechs that have clinical stage assets going through phase 1 to phase 2, phase 2 to phase 3. So we are not that accurate predicting phase 3 outcomes. But phase 2 outcomes, when the trial is for a small molecule, targeted, first-in-class, single-agent therapy, we do really well. So about 20% of the trials that we look at are predictable with our algorithms with reasonable accuracy, and I'm talking about 87 to 92%, very high accuracy. And we use 2 scores to evaluate the probability of transition from phase 2 to phase 3. So one is target choice score. So we try to implicate the target in a disease and then look at how well did the biotech company conducting the trial select the target. And then we look at how heterogeneous the target is in the patient subpopulation. So we utilize a few modules from Pandaomics for that, and also some modules that were trained specifically on clinical trial data. And then, so that's one score. And then another score is clinical study design. So there we utilize a generative approach. We use a transformer neural network that can extrapolate the trial into the future and also tell you the probability of success of that specific trial based on clinical study design. And that system is also interpretable. So we can see which features are more likely to contribute to the probability of success or failure of this specific trial. So, and we turned the system originally, it was one of the oldest projects in Insilico. It's 6.5 to 7 year old. The first paper in this field we put on archive, or just on expression data in 2016. But then we approached the problem from many angles, looked at many different approaches on how to score trials, and performed quasi-prospective validations, right? When you train up until 2015, test
Right.
2015 to 2020, for example. So when the system did not see the trials or even traces of trials, because it's trained on everything before those trials even started. And then we realized that is not enough. We need to train on, we need to produce prospective validation. So we train on everything up until today. We
Right.
make predictions, and then we wait for 1 or 2 or 3 years even, or 5 years, and wait until our predictions, the clinical trials were predicted read out, and see if we did it well. And now we are getting a lot of this prospective validation. So we started that again back 2016. We can find some of our papers on archives on preprint servers where we can date stamp, right? And there is a prediction, and we cannot remove them from there to see how well we did. And in most cases, we did extremely well. So after we validated for ourselves, and also we have virtual portfolios on Yahoo Finance and other resources showing that we can even outperform biotechnology analysts using this tool in terms of APR, for example. If you just invest, say, $1 million into a equally weighted portfolio of trials that are scored using our algorithms, gives you, I'm not going to make claims here, but it gives you decent returns.
No, I've been looking into this. I mean, it's, A, you can have a better probability of prediction, but B, you can actually tell the person doing the trial how to make changes to improve the trial, to improve the probability of success, which they may not consider because the machine is sort of looking at many different parameters to help you improve the outcome.
Exactly. And in my case, what I do also now, I try to bridge Insilico and PandaOmics. So those two systems can actually be combined for better target discovery and target validation. And now those two systems run my fully automated robotics lab. So I've got a fully automated robotics lab in Suzhou. It's an entire floor full of robots that are interconnected, and humans do not go into this lab. You just throw in a sample, it gets processed, and if you like, I can tell you about how it does it.
We're going to get to that in a second.
The data is fed into PandaOmics and Insilico. Those systems select the target, and then the system does what it does.
So let's step out of this for 1 second. There's been a huge uptick of new companies, founded, the amount of venture capital raised around AI for drug discovery. I mean, it's starting to become very blurry. I mean, you founded Insilico in 2014 before that really big wave got rolling. So arguably you were ahead of the game, right? But in past conversations with the media, I think you've predicted there's going to be a shakeout, a consolidation in the area, that a lot of these companies may go away. I think in 2021, you told Max Gelman at Endpoint News that there was going to be a shakeout in the next, say, 12 to 24 months. And if you run the clock forward to today, that 12 to 24 months puts us right about now, right? So let me ask you a few questions. Like, do you stand by that same prediction? If you had to, what would you change about it? And if AI in pharma is in a kind of investing bubble, what do you see as the fundamental weaknesses that are going to get exposed, or the assumptions that are going to turn out to be wrong? For example, are there just too many Me Too or copycat companies that don't have valuable original ideas about how to use AI in drug discovery? Let's start there.
So sure. So first of all, I think that my prediction came true, and much earlier than we expected, because in 2022, that was a real shake-up year. So several of our, not competitors, I actually don't treat anybody as a competitor, unless you are in the same target space, right? Because if you have your own discovery programs, usually you if you are in the same target space, then you're a competitor. But if you are not in the same target space, you're just competing for attention, and we never had too little of it, right? So many companies have collapsed. They went out of business, and currently the market has shrunk dramatically. So, and the trend continues. So you saw, so Endpoints just recently published an article that, well, one of the companies in California, I'm not going to say which one because I actually like the guys a lot. They just downsized, so cut 30+% of their R&D of their staff, right, and it's all scientists. So now they focus on drug development, so trying to get some of their drugs that they already discovered using AI into the clinic, right? And that's a tough choice, right? So you basically move away from being an AI company to becoming a biotech company. That's the choice that we avoided by having two CEOs, back way then, when we realized that technology moves faster than biology, than biotechnology, and many companies went out of business completely. So some went bankrupt, most downsized, and I have not seen many being created, right? So if 2020, 2019, 2020, that was the peak when huge numbers of AI companies were created, many of them are not around anymore. And there is a good reason for that. So first of all, if you are to partner with a pharma company today, you need to have a very high standard for what you're showing to them. Mm-hmm. Because most of them, they have internal AI groups that have ramped up, that know that what they're doing, right? They will look at your algorithm and they will either copy this algorithm and say, thank you very much, we didn't know that it works, right? And that happened to us many times when we were kind of smaller and less experienced. Or they would give you a pilot, and that pilot will need to result in certain, synthesizable and active molecules, preferably that could be progressed into a working program, right? Therapeutic program. So most of the pharma companies are now smart enough not to partner with AI companies that will not be able to deliver and that will not be around, 2 years from now. So it's very stupid for many pharma companies to partner with, stage Series A companies, right? Because the probability that they will be able to raise Series B and continue supporting this big pharma is very low, and became lower because we're also in a biotech winter. So many investors in AI are shying away from that. And that's the second argument. So one of the main reasons why many AI companies are dying is because investors either ask for much lower valuations to put the money in, because they understand, okay, worst case, they will be a software vendor. And the software market is tiny, right? So actually the platform market is not huge. I would say it's, if it's $1 billion globally, I would be very surprised. I'm talking about like pure AI-powered drug discovery software. Many investors would give you very low valuation. Many of them, they will bring very experienced analysts, and they will scrutinize the technology, and it will be much more difficult to fundraise because most of the time we have already had a footprint in that technology because we've explored very broadly, and several other companies as well, and some are exploring those right now. So that's second. Third is both investors and pharma companies and even employees, potential employees, like to see clinical stage assets from your AI engine. If you don't have some that tangibly came out of your AI engine and are in phase 1, phase 2, they don't believe in the company very much, right? Because again, they understand it's the biotech winter, job security or the company's future is very murky when the markets are not flying high and the investors and pharma are much more educated. So right now we see a lot of collapses. And I think that this trend is going to continue. Like, for example, one of the brightest young scientists I met recently, who did his graduate work at a very top university in a top lab, has really amazing ideas. He was asking me for guidance and asked Alex, okay, well, how do I build an AI-powered drug discovery company today with this and this and this approach? And I actually want to combine target discovery and small molecule discovery with this system. And I want to fundraise. And I told him, well, you know what? For you, specifically with all your background, I would recommend a completely different approach, right? So don't go discovery. Don't go too early. If you think that you can fundraise and sell this idea to investors, you should use your technology to in-license phase 2 or phase 1 clinical stage assets that are demonetized today, right? So there are many companies in biotech that are collapsing, that have very little cash. So go and try to work with their assets to either repurpose them or pick the best ones, and then for a specific indication and in-license, fundraise and in-license and take them forward, right? And take them through one stage. And that will demonstrate to the world that your algorithm works, at least on the target discovery side, because you can, do a better job than those biotech companies that have those trials. And you shouldn't go too early, right? Because market for software and services is tiny, and the impact you are going to make on the field is going to be very small, because you are going to be struggling and trying to crawl out of this early preclinical swamp, and most of the time you're going to fail, even if you fundraise, even if you try to do a proof of concept. But if you demonstrate that you can do a very good job with a clinical stage asset, yes, you will require more capital. Your next big thing could be the early stage drug discovery, right? You can then say, okay, well, I have validated this, and let me go back to the drawing board and now show to the investors that with my own internally generated funds, after out licensing a repurposed or progressed phase 2 asset, phase 2 to phase 3 or phase 1 to phase 3, and with a commercial success, I can now fund my hobby, which is early stage drug discovery, because it's gladiatorial there. It's very difficult to crawl out, and we would need to see another hype cycle of hyper investment to see another wave of AI-powered drug discovery companies.
I mean, I do see pockets where there's some unique approaches that I've liked. Like I was just talking to one in RNA structure and their approach, and so I think there are some paths, right? But I think that some of the well-trodden paths may be more difficult. But I do believe that because of these technologies, there are some unique avenues that have also opened up that most people may not consider right off the top of their head, that are possible, that are still very investable. But that's just, as an investor, like I've got to be looking for those, needles in the haystack to be able to get that next opportunity.
Yeah, but again, I don't want to discourage anybody, right? I'm just being a realist.
Yep.
And I don't want to sound like one of those people who criticized our tech, right? When we just started, showing some early proof of concepts for generative AI in chemistry or biology, or both. They were, of course, sounding very discouraging for no reason, right? Just because they were jealous. In this, or I don't know for what other reason, or they wanted to stay relevant. But yeah, I don't want to be one of those. So yes, start a company, great. Try to make it, but my recommendation would not to do this in traditional space, so to speak, traditional protein targets plus traditional small molecules. Go RNA therapeutics, go some other therapeutic modalities, because pharma also needs new ideas in general, not only just acceleration of small molecule drug R&D.
Yep.
Cell therapy, yeah.
So, and the last thing I want to touch on, you brought up a little earlier. You're investing a lot in this sort of fully automated wet lab where robotics can handle all the experiments you need to generate sort of larger data sets and validate your drug targets. I think you said you're building a prototype lab in Suzhou, China. I'm probably not pronouncing it correctly.
Yeah, Suzhou.
This is a theme I've heard from many companies, right? It seems like robotic wet labs are the next big thing. What's your goal for those facilities, and what's Insilico's unique philosophy about how they should run?
So sure, you could actually put those labs into two categories. One would be biology and another one is chemistry, right? So very easy. So some companies are focused on chemistry automation, and there are many. And there you should start with large contract research organizations like, Wuxi Aptech and others. They also have automated labs, but it's chemistry. So let's leave chemistry aside for a second. I think there's going to be a big revolution there, but we're not there yet, but we're getting there. In biology, we already see a revolution, right? So, and that's where I would say barriers of entry are going to be much higher than an automation of chemistry. So for target discovery, there are several generations of labs, laboratory robotics. So high throughput screening has been around forever. So since late 90s, companies started introducing those massive liquid handlers that pipetted in an automated way the molecules onto a variety of cells and cell types, cell lines, organoids, and looking at the phenotypic effect, right? So if something worked, they prosecuted that molecule. So let's call it first generation. Second generation is that they started looking closer at the data and started generating omics data, including in cases where the compound did not work. So third generation, it's more automation and more automated islands where people started integrating, for example, omics and imaging, but mostly put large imaging machines together with high throughput screening machines. And companies like, I don't want to single out some companies. Yeah, I'm not going to talk about companies, but some companies that basically are publicly traded today, they started back in those third generation era, and they focused predominantly on imaging and did some omics, automated workflows, and they have some clinical stage products right now. Fourth generation was when companies built predominantly by investors who started, getting into this field and trying to build companies with the purpose of selling them to big pharma later on or taking them IPO. So that's not organic growth. I call it, investor build companies. Mm-hmm. And for the purposes of accessing financial markets or selling. So their story was that we don't have enough high quality data. We want to generate more data using robotics. And once we generate this data, we will find new targets. And then we will also train better AI. But currently, we lack high quality data. We don't have it. So, build it and they will come, the targets. And there are a few companies that do that. I don't want to single out anybody. But I haven't seen any results from those companies yet. Our approach was very different. And also in those fourth generation labs, I call it fourth generation, you still see a lot of humans walking around with microplates and shuffling those microplates into the machines, or doing a bunch of work manually. So fifth generation lab, and that's something that I think we've achieved in November last year, or even October last year, is full automation. So where you create machine learnable data, but there are no humans in the lab at all, right? So you basically throw in a sample and the robots do the rest. So they can do imaging, all kinds of omics analysis, and then do high throughput screens, and then again, imaging and omics analysis. So we had that last year, and the sixth generation is when you actually are an AI company, a real one, organically grown. Usually you don't have clean machine learnable data coming from the robot, and I actually think that it's the wrong approach to start with. But you struggled over time and built an AI system that can discover targets without the robot. So you have enough data in the system already to be able to come up with target hypothesis yourself, right? And actually with many, many different approaches. And you battle tested the system, and then now you want to take the system to the next level. You want to automate many processes to actually make your AI-powered drug discovery faster, and also remove additional bias and do it 24/7. So that's what we did, and that's our sixth generation lab. And in sixth generation laboratory robotics, AI is making decisions on target choices. So it's already pre-trained. So you have pre-trained AI that sits on top of the application layer, where you have multiple AI models that come up with target hypotheses, and based on the data it's exposed to, it picks target, also looks at whether the molecule is already available for this target, and selects this molecule for testing. Or if there is no molecule, it can try to generate a molecule and ask us to synthesize it, right? Or it does a CRISPR screen, right? Just to see if the knockout of this particular gene will result in a desired phenotype or expected phenotype. So we have now achieved sixth generation. At least that's what I believe in. So because the devil is in the detail, and unless you show a phase 2 asset coming from this lab, all the claims are hypothetical, right, as we know, anything drug discovery. So we've built a lab, and that allows you to fully automatically identify targets, validate them without any human intervention. So you can put it in manual overdrive and select targets manually and do it also remotely. But our idea is to have a pre-trained AI that is exposed to incremental data coming from the lab. It makes decisions based on that incremental data, and then it performs some action. For example, selecting molecules from libraries, doing a high throughput screen, and then doing, again, many omics and imaging experiments to see whether the hypothesis was confirmed or not. And also you can learn a lot of biological insights from this exercise. So our lab allows you to do, sample goes in, you do deep phenotyping. So you understand, you have imaging, you have fluorescence contrast imaging, you get transcriptomics, so RNA-seq, you get methylation, you get whole genome, plus you get a few other data types that I cannot talk about. And then the system takes this data, makes decisions on the targets and on the compounds, runs a high throughput screen, takes the compounds from the storage, and then selects the cells that it wants to run through an additional screening. And you get, again, high resolution imaging, so deep phenotyping, contrast imaging, fluorescence, and you get transcriptomics and methylation and additional data types after a high throughput experiment was performed.
And so how, if you were to guess, how much faster would you think the system would then be able to produce something? And then would there be a cost differential? And, give me some hypothetical metrics, because I'm not sure if the system has produced anything yet, to put it into context of how we do things manually.
Yeah, so it produced something and a lot. It produced a lot. Currently, to run the system, it's of course much cheaper than running it manually, right? So if you were to go through the entire workflow, you would need a lot of scientists who are going to be working on these problems, and most likely they would have some inconsistencies. I remember standing on the bench myself, right, and pipetting into 96 well plate. Even if you do basic PCR, very often you just forget whether you, pipetted the reagent in the well or not, right? Did you mix? Did you not? Well, you just forget about it, right? Especially if there is, some distraction. And I think that currently we've demonstrated that we can automate the process and cost down at least by removing humans from the lab. However, the amount of data we generate from every experiment, from every run, is infinitesimally larger than what you would expect in a normal human-driven lab. And that makes it also very expensive, right? So in a high throughput screen, very rarely you do RNA-seq, for example, for every sample in the plate. You would only do it selectively for, samples where the compound resulted in certain morphological cellular change, right? That are traceable, the changes that are traceable through imaging or naked eye. And then you perform those expensive omics experiments. In our case, we actually are hungry for data, even for samples where the compound didn't work, or it caused something but not what you expected. So our cost per run is actually much, much more expensive currently than what you would do in a traditional manner. But if you are to look at the data economics perspective, if you were to generate the same amount of data manually, it would be, we are cheaper. But if you are just to follow the traditional approach, we are much more expensive.
Yeah, but the data fed into the system to then allow it to make a better decision on the next turn is significantly higher.
You can learn.
So it's going to be higher in the beginning, but technically should be more efficient and more accurate right? So you've got to invest to get there.
Exactly. And over time, as you get and generate more data and get better at it, and again, the devil is in experimental validation, right? And specifically in trying to see if the system can pick up novel biology that has not been known before. That is the coolest thing. And so far, that's my goal for this year, to demonstrate that it works and publish. Yeah, but the devil is always in experiments.
Yeah.
So our job is to demonstrate this year, and we also want to miniaturize this lab now, cost it down, so that we can even put it, again, hypothetically, we're not doing it right now. Theoretically, you can put it in the hospital. So it can do personalized medicine, something that people do anyway, because they need sequencing anyway. But at the same time, the hospital will be able to do drug discovery and share target lists with everybody. So that would be my dream. it's a big long shot. So if I can give you a lecture on this right now, because yeah, I actually had to live in this lab for the past for a few months to build it, right? Because it's not an easy
Yes.
exercise, and you could only build it in China because we rely a lot on the on AGVs, autonomous guided vehicles. So one of the reasons why we were in Suzhou is that that's where most of those highly intelligent AGVs are made. And many of the robotics components that we've integrated, they are made there, they're invented there, they're designed there. So I think that people have a very wrong idea about China nowadays. They think about China as a technology thief, but that phase is gone, right? So it's a technology creator. And now other countries are coming to China for innovation, not the other way around. And we are one of the first companies to realize this trend, and we are trying to be where this technology fountain is most abundant. So we go.
So Alex, it's been great having you on the show. I mean, I have a bunch of questions we didn't even get to that I would love to maybe schedule another show to talk about, because I know you're at the Buck Institute and talk about longevity and everything you're doing in that space. But hopefully we can book that for a different show in the future and tantalize all the listeners with some more of your wisdom and knowledge in the space.
Sure, thank you. Would love to. And for everybody who is still listening, please do go to agingpharma.org. It's a nonprofit event, the largest industry event in the biopharmaceutical industry in aging research. It's organized by the University of Copenhagen, and it's a 5-day event at the end of August. Highly recommend it. I'm supporting it, sponsoring it, helping organize it. And it's epic because it's Copenhagen, and a great place to be. If you want to explore Copenhagen and learn about aging research and drug discovery, the place to be.
Excellent. Thank you so much.
Thank you.